Hey everyone, Dr. D here, and in this video we are going to be covering chapter 25 from our Brock Biology of Microorganisms textbook. This chapter covers microbial infection and pathogenesis. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
In the first section of this chapter, we'll be talking about human microbial interactions, starting with some vocabulary that we need to be familiar with, such as infection. An infection is a situation in which a microorganism is established and growing within a host, whether or not the host is harmed. So keep in mind that when you have an infection, it could be totally asymptomatic, or you could be sick. The organism growing within the host is necessary and sufficient to have an infection, not necessarily whether you are harmed or you get sick. A pathogen. Pathogens are parasites, microbial parasites that cause disease or tissue damage in a host. And the level of pathogenicity, that's the ability of a parasite to inflict damage on a host. If an organism is more pathogenic, it's more able to cause disease in the host. And of course, different parasites have different levels of pathogenicity. Adherence. Adherence is the enhanced ability of microbes to attach to host tissues. This is where the microbe sticks to the host tissue. It is necessary, but not sufficient to start disease. So I want you to know that adherence is something that's necessary for disease. If a microorganism cannot attach to you, cannot attach to your tissues, it very likely cannot cause disease. Here we can see microbial pathogenesis. Let's start at the beginning here. Upon exposure is necessary for pathogenesis. Here are the microbes that you are exposed to. Remember, at this point, we start the infection process. This begins with adherence. The microbe must stick to the host, either to the skin or the mucosa, followed by invasion, where the microbe invades the skin or the mucosa through to the epithelium. And then multiplication. Remember, once the pathogen is inside of you, it must be able to grow. It must be able to form a colony. Then, upon multiplication, it might produce various virulence factors or toxins in order to damage the host or spread further. Spreading further means invasiveness. This means that it can spread further from the original site and travel to distant sites, possibly even through the blood vessels or the circulatory system. And this is when the damage can go systemic. This is where systemic damage occurs throughout the body. Now, during the disease process too, toxins might play a role. The microbe might produce toxins as well. This is the disease process. Remember, the infection process is different from the disease process. Infection alone is not sufficient to cause disease. Now, there are many different receptors coating both the pathogen and the tissues where the bacteria or virus binds. So these receptors on your tissues are likely sites of adherence. These are sites on your tissues. These receptors, they are the sites where the microbes attach to the host. Adhesins are glycoproteins or lipoproteins found on a pathogen's surface that enable it to bind to the host cell. So these adhesins are responsible for binding to the host cell. Here you can see these adhesin molecules on the surface of this pathogen. And notice how these adhesin molecules bind to specific host receptors. These are proteins on the surface of your cells. And this specific binding allows the pathogen to adhere to the host cell. And remember, that's necessary but not sufficient to cause disease. Now, what are these adherence structures? There are many different adherence structures. You just saw those adhesins, but capsules qualify as well. Remember, capsules form a thick coating outside the plasma membrane. And this capsule is both sticky and contains specific receptors to facilitate attachment on host tissues. And to make it worse, capsules, such as those found in Streptococcus pneumoniae, protect the bacteria from ingestion by white blood cells. So not only does the capsule aid in adherence, it also aids in what's called evasion, which means cloaking yourself from the white blood cells, making yourself less visible to the white blood cells. Here you can see the bacterial capsule, this gooey substance, the bacterial capsule. And you can see how this capsule aids in adherence to the intestinal microvilli. Microvilli are these finger-like projections of the gut lining. Now, keep in mind, in addition to capsules, you also have Fimbriae, Pili, Flagella. All three of these also qualify as adherence structures. Yes, flagella are used for motility. However, they are also used for adherence as well. Fimbriae, Flagella, and Pili are bacterial cell surface protein structures that function in attachment. So please know that these structures aid in attachment as well. Here we can see a scanning electron micrograph of flagella and Fimbriae, and these organisms would use their flagella and Fimbriae in adherence as well. Now remember, once the microorganism adheres to the host cell receptor, at this point, the organism must be able to grow on the host. This is known as colonization, the growth of microorganisms after they've gained access to the host tissue. And of course, colonization begins at birth. This is where you are inoculated with early microbes that become your microbiota. This typically starts with mucous membranes or tightly packed epithelial cells coated in the mucus. Here we can see what we're talking about, microorganisms. The microorganisms, here's exposure to microbial cells, to the epithelial cells, mucosa at first. You see the microbes attached to the mucus. Then they colonize, they form colonies in the mucosa. Now, here's an example of a microbial community on a human body surface. An example is human dental caries. You may know of these as dental cavities. This begins with microbes attaching to the tooth surface using Fimbriae or using capsules, etc., and growing on the tooth surface in plaque. Dental caries or cavities are an oral microbial disease. After initial contact, Streptococcus sobrinius and Streptococcus mutans, these are the two most likely culprits of tooth decay, attach and reproduce and form a biofilm called plaque. Here we can see the Streptococcus and a dental plaque at low magnification and then in high magnification as well. Note the capsule here. They're using their capsule to attach to that smooth tooth surface, at which point they'll use quorum sensing to form a plaque or a biofilm, and that growth as a microbial community on the tooth is what results in tooth decay due to release of acids by the microorganism. Now, at this point, after colonization, remember, we need to invade the host and spread to adjacent tissues, or even get into the circulatory system and spread systemically, right? So invasiveness is the ability of a pathogen to grow in host tissue at densities that inhibit host function. And then invasiveness can be the spread of microbes from one tissue to the next. However, at some point, these microbes can find themselves in the blood, right? And they can go systemic. Systemic means usually microbes are in the blood, spreading from, throughout the body. And when a foreign entity finds its way into the blood, the suffix is emia, E-M-I-A. So bacteremia is the systemic, the presence of bacteria in the blood, right? So anything emia means that thing is found in the blood. Bacteremia, the presence of bacteria in the blood. Septicemia, blood-borne systemic infection. This is where you have foreign entities in the blood. If it's viremia, that would be the term for viruses in the blood. Toxemia, that would be toxins in the blood. Infection, again, any situation in which a microorganism, not a member of your normal flora, is established and growing in a host. Now, this leads to this topic of virulence. Virulence is simply any ability of a pathogen to cause disease. The way I explain virulence is I say anything, any structure or ability of a microorganism that helps it to cause a disease is considered a virulence factor. So think of anything that gives an organism the ability to cause disease is a virulence factor. And it doesn't have to be an organism. It could be like a virus as well, right, which is a small infectious agent. But anything that gives that agent or that organism the upper hand to cause disease is a virulence factor. So for example, before we move on, let me give you some examples. Adherence structures are virulence factors, right? Because think about it. Do adherence structures like capsules, slime layers, Fimbriae, Flagella, I already said capsules, Adhesins, do these things help the virus or the bacteria cause disease? If the answer is yes, it's a virulence factor. So all of those adherence structures are virulence factors, because if you took them away, they would be less able to cause disease, right? Do toxins, if a bacteria produces a toxin, does that help it to cause a disease? If the answer is yes, then it's a virulence factor. Does the capsule help evade the host immune system? If the answer is yes, it is a virulence factor. Antibiotic resistance, antibiotic resistance, does it help the infection persist? If the answer is yes, it's a virulence factor. Anything that makes it possible to cause disease can be considered a virulence factor. So how do we measure virulence? Typically, the more virulent a microorganism, the more pathogenic and dangerous it is, right? So virulence can be estimated from experimental studies of what's known as the LD50, or lethal dose 50. And unfortunately, this means the amount of an agent that kills 50% of animals in a test group. So what they do is they inject test animals like mice, for instance. They'll inject mice with varying numbers of, let's say, a bacterium species, and they will look at how many of those mice die. The LD50 is the number of microorganisms it would take to be exposed to in order to kill 50% of the test animals. And as you would expect, highly virulent pathogens, these are pathogens with several virulence factors usually, highly virulent pathogens show little difference in the number of cells required to kill 100% versus 50%. And highly virulent pathogens usually have a much lower LD50. And that makes sense, right? If a pathogen has several virulence factors and it's highly pathogenic and dangerous, then you would expect fewer of them to cause disease and cause death, right? So this is why highly virulent microbes, you don't need to be exposed to many of them to cause disease. Here we see an example of this highly virulent organism, Streptococcus pneumoniae, which is one of the agents that can cause pneumonia. And you can see here that it doesn't take many cells at all, about 100 cells or so, to cause 100% death in the mice that are exposed. So even 100 organisms is enough to reach 100% mice killed. And look, the LD50 is right here. It's right here. Okay. Notice that it doesn't take much to go from LD50 to LD100. Now, take a look at a less or moderately virulent organism, in this case, Salmonella enterica, which is, causes typhus, typhimurium. Sorry about that, the tongue twister here. But notice this, because it's a less virulent strain, it's a less virulent organism, it takes several orders of magnitude more microbes in order to cause disease. You see that? And there's a big difference between the number the LD50 and the number where you get to 100% death. You see there's a bigger spread here than with the more virulent organism. Now, what is attenuation? What is this term mean? Attenuation refers to the decrease or loss of virulence of a pathogen. An attenuated strain of a pathogen is valuable for clinical medicine because they are often used for production of viral vaccines. So think of attenuation as a microorganism or a infectious agent which has been in some way incapacitated. It's no longer able to function correctly and cause disease. Oftentimes, an attenuated strain of a virus might be used as a vaccine, or an attenuated or denatured form of a toxin can be used as a vaccine as well. So, for example, with tetanus toxin, the vaccine is what's known as a toxoid. It's an attenuated form of the tetanus toxin. So attenuation means loss of virulence. Now let's take a look at the virulence of Salmonella. Salmonella is known as a highly virulent or pathogenic organism. And we can see that it has a number of virulence factors, which we'll look at on the next slide here. Several genes that direct invasion and are clustered together on chromosome at what are called pathogenicity islands. Pathogenicity islands are clusters of genes necessary for virulence, right, for pathogenesis. Another Salmonella pathogenicity island contains genes that promote a more systemic disease, also containing resistance plasmids. So take a look here. This is your genome, your Salmonella genome. And look, this is your pathogenicity island 1, right? Salmonella pathogenicity island 1. And this one's Salmonella pathogenicity island 2. These are stretches of genes found on the genome of Salmonella. And these stretches of genes contain large numbers of virulence factors. And look at this. Here are just some of the virulence factors of Salmonella. And you can start to gain an appreciation for just how virulent Salmonella is by just looking at this list here of different types of virulence factors. So for instance, one of its virulence factors, I'll just point out a couple of these. One of its virulence factors is an enterotoxin. It produces this enterotoxin, which causes diarrhea in the host, right? That's one of its main side effects, one of its main disease symptoms or signs. It also produces cytophores for iron uptake because it requires iron for growth. It has Fimbriae, remember, for adherence. It has a virulence plasmid. This plasmid has different genes on it that aid in its pathogenicity. It has a capsule, right? Capsule antigen, which inhibits complement binding. Remember, we talked about complement in chapter 26, and complement is important in the immune system. It has a flagellum for motility, but also it has an H antigen for adherence and inhibiting phagocytic killing. It has, anti-phagocytic proteins that prevent phagocytosis. It has endotoxin. Remember endotoxin or lipid A as a component of the LPS, lipopolysaccharide layer, which causes fever. And I'm not going to go through all of these, but look, these are all known as virulence factors. And Salmonella has a myriad of virulence factors, and this is why it's so deadly and so dangerous. Okay. Now, here's some more terminology for you. We're going to talk a little bit about the compromised host. Now, in a hospital setting, what you should know is that in a hospital setting, sometimes patients can pick up infections, just by being in that environment. There's several people who are sick, who have various conditions in the hospital. So you might go into the hospital for one reason and walk out with a different infection, or come down with a different infection at the hospital. And it's so common that it has its own term, nosocomial infections. These are infections that are picked up at the clinic, at the hospital. Okay. And opportunistic infections are those caused by organisms that do not normally cause disease in healthy hosts. In the lab, we typically work with opportunistic bacterium, ones that aren't frank pathogens. They're not highly virulent strains of microorganisms, but that could potentially cause disease if given the opportunity. Now, before we move on to the latter half of this chapter covering toxins, let's go ahead and take a fun little break time with Gizmo and Wicket, see what they're up to, and we'll be right back to finish off this chapter. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's carry on our discussion about pathogenesis. Now we're focusing on enzymes and toxins. Remember that with invasiveness, this is the spread of microbes from one tissue to the next. It often requires a pathogen to break down the host tissue. And this is often done when the pathogen produces enzymes that attack the host tissue. Here's some examples of tissue-destroying enzymes that we're talking about. Hyaluronidase, which breaks down hyaluronic acid in host tissues, Coagulase, and Streptokinase, which manipulate blood clotting. Coagulase forms clots, while Streptokinase breaks them down. So let's take a look at some examples here. Here you have Streptococcus pyogenes, and notice that it's on the top of the epithelial cells here, but it wants to invade down here to the subepithelial tissue down here. Now, how is it going to get through the skin? Remember, the skin is very resilient. It's a nice barrier against microbes. But what if this microbe were to produce hyaluronidase? Hyaluronidase breaks down hyaluronic acid, which is one of the components holding the cells to one another, holding the tissues intact. It's part of the integrity of the tissue. Streptococcus releases hyaluronidase. Hyaluronidase breaks down the hyaluronic acid, and the Streptococcus are able to invade. They can invade to the next tissue over, down into the subepithelial tissue. Okay, what about the next type of enzyme? Remember, coagulase and streptokinase. Here you have some Staphylococcus aureus, which have gone in through a cut, and they are just under the skin. And normally at this point, remember, the phagocytic white blood cells, such as neutrophils or macrophages, they will come to the site of infection and try to rid the infection, try to destroy these bacteria. But if the staph were to produce coagulase, this forms a clot. And the clot walls off the pathogen. It's almost like it forms like a little wall, blocking access to the immune system cells. So here the phagocytic cells can't get to the site of infection to clear the infection. Now, conversely, here's an example with Streptococcus. Streptokinase does the opposite. If this is a clot, and let's say these Streptococcus want to spread to other tissues, but they're stuck in this clot, they can produce streptokinase, which dissolves the clot, freeing the pathogen and allowing the pathogen to spread to other tissues or into the bloodstream. Now, moving on to toxins. First of all, some terminology for you. Toxicity is the ability of an organism to cause disease by means of a toxin that inhibits host cell function or kills host cells. Toxins are produced by microbes that inhibit or kill host cells. Exotoxins are proteins that are released from the pathogen cell as it grows. There are three categories: cytolytic toxins, AB toxins, and super antigen toxins. Here you can see a list of organisms and the diseases they cause and the toxins they produce. Okay, and how the toxin works. So let's start with an AB type exotoxin. First of all, an AB toxin is made up of an active or A domain and a binding or B domain. And the example here is Diphtheria exotoxin. This exotoxin works by blocking protein synthesis. And the way it does it is by the A domain of the toxin adds an ADP ribosyl group to EF2, or elongation factor 2, which prevents its function in translation. Let me show you. First of all, let me explain what's going on here. This is the cell's ribosome, the host cell ribosome, and we all know what ribosomes do at this point, right? Ribosomes synthesize proteins. Here you can see a protein being formed. This is known as translation. And what you may not know is that your ribosomes require a helper protein called elongation factor 2. That's this little purple guy here. Elongation factor 2 aids in translation, okay? And you need elongation factor 2 to properly translate proteins. However, this is what's disrupted by the Diphtheria toxin. So let's talk about diphtheria. This is the picture of diphtheria cells up here, which are known to produce this AB toxin. The B portion of the toxin, remember, is the binding portion of the toxin. The B portion binds to specific host receptors on the host cytoplasmic membrane. Then there's internalization of this AB toxin, and then cleavage of the A portion or active portion so that it's free from the binding portion. And what does the active portion do? What does the A portion of the toxin do? Well, it finds elongation factor 2, and it will ADP ribosylate this elongation factor 2. And unfortunately, when elongation factor 2 is ADP ribosylated like this, it's not able to aid in elongation, and it actually blocks elongation, and this leads to host cell death. Now, let's talk about some other types of exotoxins. These are neurological exotoxins. That means that their activity is on the neurons. The two we'll talk about are botulinum toxin and tetanus toxins. Clostridium tetani and Clostridium botulinum, these produce potent AB toxins that affect nervous tissue. Botulinum toxin consists of several related AB toxins that are the most potent biological toxins known, while tetanus toxin is also an AB protein neurotoxin. How do they work? Let's start with botulinum toxin. Here we can see the normal muscle contraction. Here you have a motor neuron. Here you have a muscle. And when you want to, let's say, flex your muscle, you want to contract your muscle, your brain will send a signal to the muscle to contract. And the way this works is the axon of the muscle will produce acetylcholine. These little A's represent acetylcholine, which then binds to acetylcholine receptors on a normal muscle, and this triggers contraction of the muscle. Okay. Now, with the botulinum toxin, that's this purple thing here, the botulinum toxin blocks release of acetylcholine and it inhibits contraction. So you see, the toxin binds to this axon, preventing the release of acetylcholine upon excitation, and the muscle remains flaccid. And this is a phenomenon known as flaccid paralysis. Botulism causes flaccid paralysis. And this type of toxin is prevalent in canned goods. You can see botulism in improperly canned foods, but it's also used in cosmetics, or cosmetic medical procedures, because this is the same toxin that's purified for injectables in the form of Botox, right? So when people get Botox in their foreheads or in their face, this is the same toxin, and it causes this paralysis where you can't, frow your eyebrows, and you can't frow your forehead and cause those wrinkles there, right? Isn't that interesting? Now let's explore the tetanus toxin, which has the opposite effect of botulinum toxin. Let's look at figure A here, which is normal relaxed muscle. In figure A, muscle relaxation is normally induced by glycine. This capital G is glycine. Glycine is released from inhibitory interneurons. So here you can see this is an inhibitory interneuron. When you want to relax your muscles, the inhibitory interneurons will release their glycine, and then glycine will bind to this motor neuron. Glycine acts on motor neurons to block excitation and release of acetylcholine. Remember, the motor neuron must release acetylcholine to trigger muscle contraction. Well, the inhibitory interneuron will release glycine, and that prevents the motor neuron from releasing its acetylcholine, and this leads to muscle relaxation, right? Muscle relaxation. So what does the tetanus toxin do? Let's talk about figure B here on the right. Tetanus toxin binds to the interneuron to prevent the release of glycine. So you see here, this is where tetanus toxin binds. It prevents the glycine release from the inhibitory interneuron. Now think about this. If you cannot release that glycine, then you can't block the release of acetylcholine, and this causes irreversible contraction of the muscle and spastic paralysis. So in this case, the muscle is perpetually contracted and you cannot relax your muscles. So when people get tetanus, if you're not vaccinated against tetanus and you come down with tetanus, this means that every muscle in your body contracts at once. And so people, their arms curl forward, curl up like this, their back arches back, their jaw clenches. Okay, and sometimes you can, this is known as lockjaw. This can lead to broken teeth and just a terrible, terrible number of signs and symptoms there. Now, what are enterotoxins? Enterotoxins are, these have effects on the intestines, right? The small intestines. Anytime you see this prefix here, entero, just think guts, right? Or GI tract. And enterotoxins are exotoxins that affect the small intestine. Generally, what they do is they cause massive secretion of fluid into the lumen of the intestine, which can result in vomiting and diarrhea. Think cholera, right? Cholera toxin. So those were all AB toxins. Now we're moving on to cytolytic exotoxins. And what cytolytic exotoxins do is they work by degrading the cytoplasmic membrane integrity, causing cell lysis and death. So for example, in lab we talked about hemolysins. Hemolysins would be exotoxins that lyse red blood cells, for instance. Staphylococcal alpha toxin, it kills nucleated cells and lyses erythrocytes, red blood cells. Here you can see these hemolysins at work. This is a blood agar plate, and you can see the Streptococcus pyogenes colonies growing on the plate. And notice how we have beta hemolysis. We have clearings around these colonies. That's because of the alpha toxin. The alpha toxin, which is breaking down their erythrocytes and clearing this blood from the immediate area. How does it work? The staphylococcal alpha toxin is a pore-forming cytotoxin. As the cells grow, the Staphylococcus release these monomers, these protein monomers. Then these monomers are 7 identical monomers will enter the plasma membrane of the host cell, at which time they oligomerize, which means they pair up. 7 of these proteins pair up. 1, 2, 3, 4, 5, 6, 7. And when they oligomerize, when they pair up in the plasma membrane, they form a pore, right? This forms a pore. The oligomer forms a pore, releasing the contents of the cell. And you can imagine if the cellular contents are just being released and also water is able to flow into the cell, this would lead to cell death and lysis of the cell. And lastly, the last type of toxin superantigens, right? Superantigens. Superantigens they cause an overstimulation of the immune system, which can lead to shock and death, generally due to localized infection, but with systemic effects. So here, superantigens, they involve the immune system. They can overstimulate the immune system, and this can have adverse effects, really deleterious effects on the host. Now, moving on to endotoxins. Remember that there's only one endotoxin. I've spoken on this before. The only endotoxin is lipid A, right? It's the lipopolysaccharide portion of the cell envelope. It's actually just the lipid portion, right? It's lipid A is endotoxin. It's generally less toxic than exotoxins. Remember, exotoxins are proteins. Endotoxin is not a protein. Endotoxin is simply lipid A. It's the lipid portion of LPS. So it's only found in gram-negatives because only gram-negatives have an outer membrane, right? And the LPS component of that outer membrane of gram-negative cells, it includes lipid A, endotoxin, okay? By the way, if the presence of endotoxin in your blood can be detected via an assay called the Limulus amoebocyte lysate, or LAL assay. The presence of endotoxin can be detected by this LAL assay. And what's cool about this assay is that we get the reagent for this assay from a horseshoe crab, right? We bleed horseshoe crabs, and their blood is used in this assay to spot endotoxin. It's pretty kind of a fun fact for you. But again, what is endotoxin? Endotoxin is quite literally a component of the outer membrane of gram-negative cells. Remember LPS, which has, the lipid portion, the core polysaccharide, and then the O side chain, the variable O side chain. Well, the lipid portion here as denoted as a yellow box, the lipid portion of LPS is endotoxin, period. That's the only endotoxin. And how do endotoxins vary from exotoxins? I want you guys to know this. Endotoxins are resistant to heat. They're heat stable. Think about this. An endotoxin is a lipid. Lipids don't denature in heat, okay? So you, for example, if you have food and it's contaminated with endotoxin, lipid A, you can't just heat that food or cook that food and expect it to be now safe to eat. If it has endotoxin in it, it'll still make you very sick. So I want you to know endotoxins are heat stable, while exotoxins are heat labile. They are susceptible to heat. Why? Because exotoxins are proteins. So, and by the way, exotoxins are proteins, which means that they come from genes, right? The microbe has the gene. The gene undergoes gene expression to make the exotoxin. Okay, so every exotoxin is produced by the exotoxin's gene, whereas endotoxin is simply the lipid portion of the LPS, of the outer membrane. Now, endotoxin is lipid A from gram-negative cell walls, whereas exotoxins are proteins, and there are so many different exotoxins. Endotoxins are weakly immunogenic, while exotoxins are highly immunogenic. What does this mean? This means immunogenic means can elicit an immune response, right? The immune system is alerted, right? Endotoxins are not very good at eliciting an immune response. They are weakly immunogenic. Exotoxins, mainly because they are proteins, and proteins are so easily identified by the immune system. Exotoxins are highly immunogenic. Here you can see a table summarizing the properties of exotoxins and endotoxins. And that leads us to the end of the chapter. What a fun chapter, really interesting concepts. Hopefully it made sense. Let me know if you have any questions in the comment box below, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D,